And the reason why I'm picking on 314, 315, actually, we're going to kind of extend this all the way out to like page 318, 319. And everything that you really need to know is going to be found on this slide right here as far as replication goes. Okay. But there is a lot going on. And to where it's kind of nice to see things at least broken down step by step by step by step by step. And that's kind of what we're seeing with 314 and on to like 315, 316, and just kind of all this stuff is drawn out for you in a little bit more step by step format versus just everything right up here that we're looking at. Okay. So the ultimate goal that we're hammering out today, because yesterday, if you remember, let's go this route. Yesterday, if you remember, we talked about the structure of a DNA molecule. Okay, and we gave you this term of anti-parallel. Okay, and so somebody in third period today, I'm like, well, what's anti-parallel mean? Well, it's not parallel; it's perpendicular. Which, I mean, you could definitely assume that from the word itself, right? Okay, but that's not what they're referring to here when they talk about anti-parallel. And again, we drew this up yesterday. But if I take and I split that, okay, that the DNA molecule, the sides are still they're parallel with each other, right? But the whole anti again comes in the form that one is running one direction, the other one is running the opposite direction. And how do we know that? Well, it's based on these sugar molecules, these pentose sugars, and which way that they are shaped or which way they're pointed, or simply where that 5' and that 3' end are, right? Okay, so for example, if I go to this molecule over here, and I know you guys have that drawn out or written out already, okay, but if I go to that molecule right here, I know that this is the 3' end because my third carbon or that third carbon is on top, okay? I know that this is the 5' end because that fifth carbon is on the bottom, right, of that strand. You guys see that on that molecule, okay? Because today it's going to get brought up, and it's going to be complicated. Right? I'll tell you that right away because I'm going to be talking 5', 3', 3', 5'. If we don't understand that basis, right, it's going to be tough to kind of grasp all of this today, which we're not just going to be done with it today. We'll continue on with it a little bit tomorrow and also on Friday. And then again, the whole anti-parallel is that, again, this would be the 5' end over here, right, and this would be our 3' end. So we can say that this molecule is actually working up, right, and this molecule is working down, which those arrows have already, shown you, okay? Any questions on that before we move on? McKinley, what do you got?
So there's something I'm missing. Just because, like, on one side you have 3' and 5'.
Mm-hmm.
I don't understand that.
What do you mean on one side? Well, even when I look right here, I still have a 3' carbon right there, right? Every one of these sugar molecules is going to have all 5 of those carbons. Okay? Same thing here. I've got 1, 2, 3, 4, 5. I've got 1, 2, 3, 4, 5. They're there, but it's all in relationship to where that fifth carbon and that third carbon are. Okay, so even if I look at one molecule, I know that my top is going to be 5'. I know that my bottom is going to be 3', right? If I look at this, if I take this molecule right here, all I have to look at is that one. I can see, well, my 3' is up here, my 5' is down there, my top is a 3' end, my bottom is a 5' end. Okay, is that you're good there? Okay, all right, yeah. So I mean, again, you just look at one sugar, and it tells you. It's going to tell you what end is what. All right, and if you can figure out one side, you automatically should never assume anything, but you can assume what the other side is, right? Okay, all right, okay. So now here's the trick part. Right, we're going to talk replication. Okay, and we started today. We're going to start today with just a little bit talk bigger goals, bigger pictures. Right. So if we're talking like what is replication? Like what is happening during that process? What are we cloning? Big picture. Yeah, we're looking at cloning. I think replicate, I think a copy. Right. So what we want to do is we're gonna sit back and our cells are gonna sit back and they're gonna copy DNA. Okay. Now I always like to talk about goals, right? And what's the goal? I mean, why is the DNA copy? Like, why do our cells need to make sure that is happening? Yeah, okay. I got that part. What do you got? You were going to answer my question. How didn't you know what I said?
No.
Oh, what did they say?
Yeah.
So the cells can split. Yeah. So cell division, right? Okay. So ultimately, when you look at it, we want to basically, we're going to split, we're going to duplicate, we're going to double the amount of DNA so that when a cell divides, right? We ensure that every cell has the exact same copy, unless what happens? Mutations occur, right? Heck yeah, I do. Look at that. That's good stuff. Thanks. Appreciate it.
No.
That's good stuff. Okay. All right. I like food if you can't imagine. So again, so we talk goals, right? So the goal of this again is to ensure that when a cell divides, and I'm just going to put down cell division because that's what it comes down to, is that basically when a cell is getting ready to divide, right, that ultimately it's going to ensure that every cell has the exact same copy of DNA, right? And when we'll talk mitosis, meiosis later. Here we're talking again this overall process of mitosis, because in meiosis, right, we still see replication happening, we still see the S phase happening, but in that case, we don't really care so much that the DNA is the same. We want that to be completely different, right? Yeah, we want that to be genetically diverse as we possibly can. Okay, now let's take a step from there. If we're talking cell division, okay, what would be the reasons on why cells divide? Why would cell division need to occur inside the body? Yeah, one, we've got death, right? Exactly. Okay, cells in this case, and I'm also going to throw in like damaged, right? So if you got cells that are dying, apoptosis happens, something, basically they're marked for death, whatever the case is, right? So we start looking at that as being a reason, right? To regenerate more of them because they're dying or they're damaged. Okay, good. What else? Yeah, absolutely. Okay, probably the biggest one is going to be, again, when cells get too big, they're not very efficient, right? Surface area to volume ratio gets too large. Therefore, we know that they've got to divide. Okay, what's the other? I mean, there's probably one more, and it's not even a stretch. But what would be another reason on why cells are dividing in an organism?
To grow.
Yeah, to grow. Absolutely. Okay, growth and development. Right, I'm done. I'm on my downhill slide, right? Okay, but you guys, for the most part, probably not all of you, probably some of you have hit your growth spurt and you're done, right? Creighton, for example, a little 4 foot, 50 pounder. I'm hoping he's still growing, okay, all right? And he's probably going to put, but again, what's happening? He started out, all of you guys start out as one cell, right? Sperm fertilizes the egg, creates a zygote. That zygote, by the process of mitosis, splits. You get two get to four, and eventually makes you guys, right, or makes us, right? So again, three goals. I shouldn't say three goals, but three reasons on why cells are divided. Okay. Now, the other part that we need to get in the case of replication is this process that they call a semi-conservative model. All right. And it's more of a big picture of replication happening. And we're going to get in depth and we're going to talk about this process. But again, it's going to be more about what the bigger picture as far as what's happening. So understand that there's like probably 7 or 8 to 10 different ways that we could draw out a molecule of DNA. Okay. One, I could break it down structurally just like this right here. Right. I could have shapes. I could draw it. Even then, this is probably not specific enough because I could draw out every single element, right, where it's found and everything else, right? But then I can also make it relatively broad. And when I'm talking about broad is that I could also just show a strand of DNA as being something like that, right? And the two lines are representing basically just the two sides, nucleotides. This nucleotide or these groups of nucleotides over here and these groups of nucleotides over here. So the whole point behind the semi-conservative model, right, is that when replication happens, okay, is that basically this molecule is going to unzip, right? We're going to use that term unzip, and I'm going to put little quotes around it because it's not exactly what's necessarily happening, but it's going to unzip, right? And then we've got enzymes that are going to come in and they're going to base up, they're going to pair up nitrogen bases, right, with each strand. So what we end up getting is that. I even hate to even do this because I don't know if it's going to come back to me. And if that's the new strand, that's what we get with the semi-conservative model. Okay? And so what it's referring to that in the new strands that are being built, okay, we have some old and we have some new. Okay? Now, what would be the advantage, again, of having some old and having some new in these molecules? Less chance of mutations. And why is there less chance of mutation? Because again, when these are splitting, right, we've got an enzyme that's going to come in. It's going to read this half over here. And as it reads it, it's going to pair up the proper amino acid or proper nitrogen bases, right? Same thing over here. So there's a lot better chance of no mutations happening when it's got that template to simply read directly and attach nitrogen bases to it. Okay, so that would be, again, what we consider to be the semi-conservative models. And there was other theories out there in the past, nothing that we're going to hammer out. But again, this is the model that they have come up with. And it's a model that basically they figure is happening during the process of replication, or is happening during the process of replication, to ensure, again, that each cell is getting, as it splits and divides, the exact same copy of that strand. Okay, now on the board, what I have is I have a list of terms, and actually what it is, it's kind of more of a flow chart, okay? It's going to take you basically step by step by step, right, kind of through the overall process, as well as tying in the enzymes and where they come into play. And as you guys go through and read like the topic 6.2 in replication, right, again, at the bottom, it's telling you the ones that you need to know. DNA polymerase, it's up there. Ligase, it's up there. RNA polymerase, we'll talk about that. It's basically the same thing as DNA polymerase, but more obviously directed toward RNA. Helicase, and then this topoisomerase. Okay, so we've got those going on, as well as simply just what's happening when this process is occurring, because it is way more complicated than just saying that it unzips, enzymes come in, new nitrogen base is attached, boom, we got a new molecule, we're good to go. Okay, so the first little bit up there that I'm going to do, and actually, again, this starts out on page 314 with you guys, okay? And the whole point of me looking at the book is, again, because it's taking you more of that step-by-step kind of production. And the first part, actually, we can go through and take a look at is going to be 314, 315, and so on down the line. And the first word that I have up here is called origin of replication, okay? So if I bring up the term origin, what does origin mean? Yeah, the beginning, okay? So it's a start of replication, okay? Now, one of the myths, or one of the things that we never talk about in bio, right, is the fact of, we always talk about the unzipping of this molecule. We always talk about how it straightens out, right? And then there's this unzipping, and that allows enzymes to go in and simply match up. But what we start looking at is on every strand of DNA, okay, there are, obviously we know introns, we've got exons, we've got junk DNA, we got DNA that needs to be copied off and everything else going on, right? Okay, and every one of it is relatively valuable in its own little shape and its own little way, okay? But strung throughout, there are these points of origin, okay? It's not like the molecule starts at the top and it just unzips, right? Because one, if it did, and we're talking relative time, there would be too much time, right? There would be too much energy taken for simply one enzyme to go through and replicate this whole strand by itself. So power by numbers, okay? And so what the power by numbers basically falls into is that in every so often, there's going to be a certain DNA code, right, that is going to be the point of origin, right, or this origin of replication. And again, it could be right here, it could be down there, it could be down there. And the whole point of this is that replication doesn't simply start at the top and work down to the bottom. It is going on continuously, and it's going on almost everywhere on this chromosome at the same time, okay, or on this DNA strand. Does that make sense? Okay, so enzyme's going to come in. It's like, yep, boom, we're going to open it up right there. Enzyme's going to come in down here. Yep, we're going to open it up right there. It's going to move on down. We're going to open it up right there. And remember, we're talking about, I think, even on chromosome 1 itself. Okay, remember, chromosomes are made up of DNA, right? We're looking at 279 million base pairs, right, that are found specifically on chromosome number 1 itself. So again, in order for that to start to top down, it would actually take a long time. So all these, there's multiple points of origin throughout that strand to where replication is occurring at the exact same time. Okay, we good there? Okay, so looking for a special code. Now, what this causes, because what's going to happen is at that point of origin, right, there's going to be enzymes that are going to go in, and they are going to break the hydrogen bonds. Remember yesterday we talked about how DNA, relatively weak hydrogen bonds on the inside, right? It's relatively stable because we want it to maintain its shape and its structure and such. But again, it's relatively unstable because we want those hydrogen bonds to be able to separate and break apart. Okay, so what we end up finding is that at that point of origin, it's going to start to open that strand up. Okay, makes sense to you, right? And what it causes, it's going to cause, right, a replication that's not forked. It should be a replication fork. Okay, and so when I take a look at this model that's on the board right here, okay, here would be the so-called fork. I need to get my need to get my right color up there. Go here. Here would be my fork in the road. And see how it separates those strands there? Okay, so here would be my replication fork. But another term is also going to come up, and that we can use what we call a replication bubble. Because remember, if there is a fork over here where it's opening up, on the other end, what is there also going to be? There's going to be another fork on the other end. Okay? So it's like taking a string of yarn and pulling those fibers open, right? And creating two halves. Well, what are you going to have? You're going to have a bubble there, right? Okay, you're going to have a fork on this side over here, and you're also going to have a fork on this side over here. And so when we talk about that replication bubble, it's going to look something like this. These strands would be together. These strands would still be together. And now what we're starting to see is that replication is occurring, but it's not just occurring in one direction. It's actually occurring in both directions. So now you've got these point of origins happening all over the place, right? You've got multiple replication bubbles, multiple replication forks, and they're sitting there and they will work in opposite directions. Okay, so eventually, when this continues on its path, it's going to meet up with another one, that's coming back at it. As this one is continuing to the left, it's going to meet up with another one that's coming to the right. And so again, at multiple points of origin, this happens relatively quickly and relatively efficiently because again, it's just rapidly occurring, just happening all over the place in this molecule, more than just one spot. Okay, but there's a problem. Okay, so think about this: if I happen to take let's say for example, Rambo and I, we've got a string of yarn, whatever the case is. Okay, just rope. Okay, and I give him a point of origin. Okay, he takes it. Right, he gives me a point of origin. Right, and we sit there and we start to pull, and we start to pull, and we start to pull, we start to pull, we start to pull. Okay, now we know that coil, that fork, is eventually going to move his way, right, and his fork eventually is going to move my way. But the problem with that strand is that if we keep opening, what's happening to those strands in between both of ours replication forks? Okay. The tension gets stronger. Have you ever taken a rope and realized actually you see a supercoiling process? And the supercoiling process, because like his rotation is actually counteracting against my rotation. And what starts to happen is actually starts to get tighter and tighter and tighter in the middle. Now, the actual replication forks and stuff, I don't disagree with that, right? Those are becoming loose, right? Which again, those are becoming what I would consider to be less tense. But in the middle between the two, his rotation is going against my rotation, and it's going to cause it to actually tighten up and become even more and more and more tense. A lot of times, even that tension actually causes it to wrap itself around each other. Does everybody understand that picture? Yes or no? I wish I had a visual for it. I don't have a visual. I don't have any rope or anything here.
I would say grab a string, put it through a button, when you spin it, pull it apart.
Yeah, exactly. No, I mean, I can visualize. I've done it before. I've seen it. I know what's happening there. Okay, but it's an overall process where you're going to see this super coiling that's going to happen in between both of them. Okay, so there's an enzyme that is caused that actually aids in spreading that molecule, that DNA strand apart at the point of origin. And then there's another enzyme that's going to come in, and it's going to help to reduce that supercoiling that's occurring between the two replication forks. Okay, and the only way that it can do that is if it starts to break the bonds in between individual nucleotides, and it kind of allows them to unravel and unwrap, right? And then it simply puts them back together again. Right, and so you've got all these enzymes that are working together. One of them is basically, again, opening that replication fork up or opening that bubble up a little bit at the point of origin. And you've got other ones that are going up ahead. And they are, again, kind of taking care of that tension and relieving all the stress on that molecule itself. Okay, so when we look at the term helicase, ASE at the end again means what? It's an enzyme. And if you take the word heli, right, what is that referring to, right, in the molecule? Or what is that referring to in relationship to DNA? Yeah, the helicase, right? The actual helix, the helical form of the molecule. So in this case, when we take a look, helicase is going to be the enzyme, right, that is going to uncoil, right, or simply open that little portion of the DNA molecule up, okay? But ahead of it, we've got this topoisomerase. And that is the enzyme that's going to be responsible for going ahead, and it's going to reduce the supercoiling and actually break bonds and allow that to kind of unravel and uncoil, and eventually it's going to piece them back together again to ensure that basically both, the two independent forks match up with each other. Yes, ma'am. This one is doing what? breaking it. Okay, this one is actually creating the forks. It's creating the bubble. This one is reducing the amount of stress ahead of it. Okay, so the helicase is going to come first, and as it continues and it creates more of a stressful environment because of that supercoiling, that's where this other enzyme is going to come in. It's going to break bonds and it's going to simply reduce that stress, right? And it's going to piece it right back together again, but it just ensures that that molecule doesn't, I wouldn't say self-destruct, but doesn't eventually, break due to a super amount or high amount of tension. Okay, we good there? So big picture, right? Okay, point of origin. Helicase splits, topoisomerase goes ahead and it's going to reduce the amount of stress coming from basically the uncoiling of that molecule, all right, and why that works the way that it works. Okay, we good? Yes, ma'am.
So the top, the one that starts with a T, like the green circle,
Topoisomerase, yep.
that's what uncoils the DNA? Or the blue part does?
The blue part is going to uncoil and break the bonds in between the nitrogen bases. So that's what's going to kind of cause it to unravel. But as it's unraveling, ahead of those forks, it's going to continue to get more and more and more coiled up because you've got two forks that are actually running, into each other. So that topoisomerase is going to go ahead and it's going to start to break bonds between the nucleotides, which will in turn allow them to kind of unravel as that helicase continues on. And then as soon as that tension is gone, it pieces them back together again. And now those two forks eventually run into each other and you've got a complete strand. So even though they're working independently, eventually they have to fuse together. Eventually they have to, they're going to come together, right? And that's what that topoisomerase is going to do is it's going to help to reduce the stress ahead of the game, breaks the bonds, boom, bonds reform. Now continue on until they eventually meet up with each other, until these forks meet up with each other. Good? Okay. All right. We'll get into the difficult stuff now. Okay? Yeah. Okay. So, yeah. So in this case, we're going to take a look at, like on the board, this molecule is actually working from left to right. Okay. But also understand that on the other side of that, there's another enzyme working from right to left, right? Okay. Just make sure you guys have that understanding. And if you want a good picture again of this, right, if you guys go to, basically, if you kind of look at the overview on page 316 up top, it's giving you a relatively decent picture, again, of kind of that replication bubble, those replication forks, right, and just kind of overall direction of where replication is going, okay? All right, yeah.
So that moving left to right, and this one on the other side, right to left. I'm just wondering, because it looks like it's building up the DNA on the other side with two polymers. It has the boxes on the other side. That's why I'm confused.
You got one right here, you got one right here, right?
Yeah, so it's building up two new molecules.
I'll explain that. Okay. Now, here's what we've got. So molecules being split. Well, one, when the DNA strand is split, they're independent, right? They're not together. So the one reaction is going to happen on the bottom strand. The other reaction is going to also happen on the top strand. So whatever is going on down here, it's not the same exact thing, but that's also going on up here. That's what we need to go over right now. Okay. So here's where we're at. So remember 5' to 3', right? Okay. So anti-parallel. So here what we're doing is we're going to take a look at the initial strand. Okay, we're not going to look at the ones that's being duplicated right now. We want to look at the ones that are the initial strand, and it would be this guy right here, right? So on this end, I have my 3', right? On this end, I have my 5'. Okay, if I follow that coil around, it's going to go over here, okay, which this makes this end my 5', because over here is my 3, right? Okay. Over here is my 5'. If I follow that little coil around, okay, right over here is my 3' end, right? Okay. Here's the rule. A nucleotide or a nitrogen base can only be added to a 3' end. Okay, write that down. Get to know that. And that's going to make life a lot easier, because if you can't remember that and you can't associate where the 5' versus the 3' ends are, okay, that's where the confusing part, that's where the confusion comes in on this. Okay? So we're going to start with what they call the leading strand. Okay? And the leading strand in this case, we're going to look, is going to be on page 315. Okay? So go to the 315 real quick. Okay. We're going to notice that as a molecule is built, bases are only added to the 3' end. So a molecule, a new strand, the new strand being built is being built from 5' to 3'. Okay, it's being built. Okay, but remember, because that strand is anti-parallel to the one that it is complementing, okay, the other strand is going to run in the opposite direction. So if that molecule is being built from 5 to 3, it's actually reading the original strand from what to what? From 3 to 5. Okay? So when I look at this leading strand, for example, notice, okay, here's the original strand, 3 to 5. Okay? We've got an enzyme that's going to come in. Its name is DNA polymerase, poly meaning many, polymer, it's building the molecule of DNA up, right? And it's going to read that molecule from 3 to 5 prime. And as it's going, it's going to build, it's going to attach you to it. Why? Because again, I know that this is 5 prime, right? If this is 5 prime, everything to the right of it is going to be what? 3 prime. So it just keeps attaching, it keeps attaching, keeps attaching, keeps attaching. Okay? Now we're going to go back a little bit. Before DNA can bond, before DNA can actually start attaching, right? There's something that there has to be something laid down before. Okay? And when you think about a primer, what does a primer do in terms of painting? Okay, it allows the paint to stick better. Very good. Okay. We always talk about why, if you can tint primer, why don't you just put primer down? Well, because primer is very sticky, and that's going to allow dust and debris and everything else to stick to it very easily. It gets all dirty and it's tough to clean up, right? But you put a primer down, block, stain, stuff like that. But more importantly, because it allows paint to stick to it. Okay. So the first thing that's going to be laid in this process, there has to be a primer that's put down. And as you guys see on page 316, right, or 315, excuse me, you're going to notice that there is a primer on the far right-hand side, but also notice that it is made up of RNA. Okay? So it's like a base that's like 5 to 10 nitrogen bases long. That is set down, right? It's going to join a section of that DNA. Now from there, we've got DNA polymerase that's going to come in, and it just keeps attaching new nitrogen bases to that 3' end, and it just keeps flowing, and it just keeps flowing, and it just keeps flowing in the direction of the replication fork, or in the direction of which way that fork is being opened up. Okay, why they call it the leading strand? Because it works very, very efficiently. It works really nice. It's smooth. This is actually being built in the direction of that replication fork. Okay, questions there?
So you have one side strand of DNA, and you have nitrogen bases added to it,
Mm-hmm.
and then if this bond is the 5, then you can have 3.
Yeah, if you're talking the new strand, right?
Yeah.
Yep. So that new strand, if the bottom is 5, we know that up here has to be the 3' end, right? So nitrogen bases can nicely be attached to it because they can only be attached to the 3' end, right? Okay, so therefore it just keeps attaching, just keeps attaching, just keeps attaching, just keeps attaching automatically to that 3' end. Okay, does that make sense to you?
Oh, it goes in the direction of 3.
Molecules being built from 5 to 3. Yep. So in that new strand is going to be built from 5 to 3. But it's reading the original strand from 3 to 5 because remember it's anti-parallel. Okay. So when I look at the one on page 315, that replication fork is actually moving from right to left. You guys see that on that page? Are you guys all looking at it on page 315? Okay, at the bottom, right? It is moving. I'm looking at the one, the 1615, right? And it's the one going to be on top of that. It shows the original replication, right? Okay, it's reading the original strand from 3 to 5, and it's building that new strand from 5 to 3. The little red section is what? RNA primer, right? From that now, you've got DNA polymerase that's going to go in, and it's going to attach new nitrogen bases, and it can do it very smoothly because, again, the only thing that's exposed here is the 3' end, right? Okay, you guys with me on that? Leading strand. Okay, very simple. Relatively simple. Okay, but on the bottom, and here's where the problem comes in. If I flip the page, right, and I start looking at 1616, okay? On the opposite end of this, or on the opposite side of this, you've got what they call a lagging strand. Okay, now anytime that you hear word or lag, what does lag mean? Yeah, it's slow. It's gonna fall behind a little bit. Okay, and there is a reason on why this is called the lagging strand. And I'm gonna use this. The top one up here is actually referring to a lagging strand. Okay, so my original strand is going from 5 to 3. Okay? And my replication fork is moving, in this case, from left to right. Okay? But as we said, a molecule, the new molecule can only be built from 5' to 3' end. Right? But when you look at the original strand, here's my 5', here's my 3', which means that my new strand is actually starting out with the 3' over here, and my 5' is found over here. So what's the problem? It can't build it as efficiently because what do we know? Nitrogen bases can only be attached to what end? The 3' end. So in this case, what it's referring to is that the nitrogen bases really want to be going this way, but the replication fork is going which way? It's going that way. So now, here's what I'm getting. Process called, basically they're called Okazaki fragments. Okay, now kind of come into play. And it's kind of like laying down a railroad track. Okay, you got a set of tracks, you lay it down, right? You've got another set of tracks, you lay it down, you basically pull it back, and it joins that last set, right? Okay, you go ahead of that, you lay down a new set of track, you pull back in, you join with that new track. You go ahead, you lay it down, you pull back, and you join that track. What's happening? Every time that they want to put down a new section of track, they have to do what? They've got to go ahead and pull it back. They got to go ahead and pull it back. They got to go ahead and pull it back. So what we end up seeing is that these Okazaki fragments, which is based on the guy that had found it, is that every single one of these little orange deals are considered to be an RNA primer. Okay? So if you're going to notice, what is that RNA primer doing? Okay, it's going to go ahead and it's going to set up maybe 5 to 10. I'm throwing that number out there. It's probably thousands when it comes down to it. But we're going to look at 5 to 10 nitrogen bases ahead. And then now there's a gap. And so what happens is now these nitrogen bases can get added one by one by one, and they're being added in the 3' direction. Once that's completed, we're going to have a new RNA polymerase that's going to jump ahead. Now new nitrogen base attaches. That now is joined up. It's going to jump ahead another 5 to 6 to 10 bases, right? And now a nitrogen bases can be attached because it has 3' ends that are now exposed. So it's kind of back building, right? It's still going in the direction of the replication fork, but it has to back build because the only way that nitrogen bases can be attached is to the 3' end. Okay? Leading strand works nicely in that direction because the 3' end is towards the replication fork. The lagging strand doesn't work so nicely because the 3' end is actually on the opposite end of which way the replication fork is going. So why would you consider it to be lagging? It's not as efficient, and it takes a little bit longer because there's got to be a new section of track laid, right? Yeah, road laid in order to join up. And then you've got all this all these RNA primers sitting there as well. Okay? Good? Yes, ma'am. Yeah, absolutely. Yeah. So again, when you look at the book, okay, remember you've got that replication bubble, right? So what I'm looking at is that if I have, like, let's say it's going this way, here's my lagging strand up here, here's my leading strand here. On the other end, this molecule actually is the leading strand. This one down here would actually be the lagging strand because it's working in opposite directions of each other. Okay, which again, big picture stuff to kind of realize and understand, but probably nothing that's going to come up on the exam. But yeah, I mean it's going to be going in the opposite direction, right? So again, when I go in that opposite direction, these two are actually the terms are flip flop based on how that molecule is being built during that, going in the opposite direction. Okay, right? Questions?
Does the RNA primer start the origin of replication and then work toward one end of the fork?
Yep. So you're going to have both directions, right? So you're going to find that point of origin, and there's going to be basically, again, RNA primers basically just working in both directions from that point of origin. Okay? Now, here's the other problem. Okay, the other problem is now when I take a look at the leading strand, okay, realizing there is DNA primer or RNA primer sitting there, right? And we're talking DNA. So what's got to happen to that RNA primer? Or or
Created?
Time's the wrong thing.
say it again.
Time's the wrong thing.
Say it again.
Time's the wrong thing.
It is it's wrong, right? Is that what you said? Did you say it's wrong?
Oh, I said it. Time's the wrong thing.
No. Here's what I'm looking for. Okay. If it's not broken down, it's going to be converted because the A's and the C's and the G's are already there, right? So the only thing that needs to be changed is the what? Is the U to a T. So you've got another enzyme coming in, and don't worry about that enzyme coming in, right? It's called DNA ligase, which I don't, oh no, we do need to know DNA ligase, right? And so the DNA ligase is another enzyme that's there to convert that RNA primer into DNA, okay? Which again, with the whole leading strand, not a big deal, right? Because the only part is just at the beginning. But when you take a look at that lagging strand, all right, and again, why it lags behind is because all of these steps have to happen, right? You've got the Okazaki fragments that are joined in, but then you've got this DNA ligase sitting here, and it's coming behind, and it is now converting RNA into DNA. Okay, does that make sense? So now you have a complete strand of DNA, right? Not with a mixture of DNA and RNA. Yes, Mr. Rahman.
It takes off the oxygen of the ribose sugar too then? It's deoxyribose versus ribose.
Yeah. I mean, basically, you're looking at RNA versus DNA. Yeah. Yep. So your sugars and your phosphates would be a little bit different there as well. Yep. What do you got?
Is DNA primer on? Like, because you said the DNA primer is converted.
RNA primer.
Oh,
RNA primer is converted into DNA. Yep. Remember, the ultimate goal is we're trying to make a DNA strand, right? But the problem at the beginning is you need this primer, but the primer is made in an RNA strand, in an RNA molecule, right? So it's got to get converted back to DNA in order for it to be complete, if you want to say that. Okay?
So
All right. Go ahead.
So just cut out, get rid of the oxygen, turn it back to deoxyribose, and then change the 0 cell to that.
And I don't know if it's exactly that part is that's going to cut out the oxygen.
Yeah.
Okay. More than likely what you're looking at is again, you've got your ribose sugars and you've got your deoxyribose sugars, right? So it's going to be more about converting and not suddenly just cutting off an oxygen. All of a sudden you're good, but it's converting that sugar obviously into, yeah, absolutely. Okay. All right. And again, what's doing that? Enzymes are aiding in that case, right? Yeah. They're going to basically distract one, boom, bring in the other. Now we've got DNA, right? Okay. What do you got for me? Yes, ma'am.
what's the thing under DNA ligase?
So we'll talk about that tomorrow. Okay. And it's an end of the enzyme, and we'll address why this comes into play, but they're called telomeres. And so, which is kind of neat because telomeres is actually a new research that's being done. It's actually relating or kind of a relationship to us aging, cells aging, us aging, and eventually could be the ultimate reason on why we die along, basically death that we basically are on right now, so, but we'll address that a little bit more as time goes on. All right. We're going to come back and talk about this more tomorrow because I know it's clear as mud to most